Okay, so I think I'm the first one to do it on YouTube, but this is actually very, very important because usually when they explain about mitosis, they give you a fly cell or some sort of a bug cell that contains like 4 or 6 chromosomes. But on this video, I'm going to give you all 46 chromosomes when it comes to human somatic cell, and we are going to see exactly where are the chromosomes found on during each stage. So let's start from before the S phase of the cell cycle. We are going to have during the S phase, before the S phase of the cell cycle, 46 DNA molecules. We are going to have 23 from the father side and 23 from the mother side. So we have 46 DNA molecules, which give us 23 pairs of chromosomes. Now we are going through the G2 phase and then we are going to starting the M phase, the mitotic phase. We are going to have something very interesting. During prophase we are going to start to have the condensation. But after S phase, where we pass the S phase because this is before the S phase, we don't have 46 DNA molecules. Now we have 46 times 2 because all maternal DNA molecules are going to duplicate, and boom, and all paternal DNA molecules are also going to duplicate. Copy and paste, boom. So now we don't have 46 molecules of DNA, now we have 92 DNA molecules. But how many chromosomes do we have in the start of prophase and the condensation to chromosomes? We have 46 chromosomes. Because one chromosome contains 2 chromatids, and one chromatid is one DNA molecule. So we have, for example, the maternal chromosome connected to one centromere, therefore it's a single chromosome. So we have one chromatid and 2 chromatid. This one is just a duplication of the same DNA molecule. So it's 2 DNA molecules that basically the same. And we also have our paternal DNA molecule and the duplication of DNA molecules. The chromosomes are connected through one centromere, therefore they are a single chromosome. And we are going to have 46 of these pairs. So overall we are going to have 46 chromosomes during the prophase of a human cell. But we are going to have 92 chromatids during the prophase. Now we have, and it's going to take a while me to actually draw it. Let's draw 46 pairs of chromosomes and draw them in the way that they are going to show in the metaphase plate to understand exactly where the chromosomes are going to align, how they are going to align. So we are going to have basically one. I'm going to draw it as a X because it's going to be easier. So this is, the red was the maternal, so 1, 2, 3, 4, 5, 6, 7. Let's take it and take it here, and we can keep drawing. 8, 9, 10, 11, 12. Let's take everything, make it smaller and duplicate it. Just to emphasize the fact it's so important, this lesson is so important. I don't want you guys to forget how many chromosomes we have during mitosis of human cell and during the metaphase, and where the chromosomes are actually found in the metaphase plate. So we're going to have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Now we have 24. Now we have 36. And now we have 48. So we are going to delete 2. But we said that half of these pairs are paternal. So let's say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. All of these are going to be, let's choose them, and they are going to be in a different color. So these are paternals, and we have 46 chromosomes during mitosis of somatic cell. So what do we have now? We have 23 pairs of paternal, we have 23 pairs of maternal, and during the metaphase plate, all of them are going to come together and align on the same plate. So we are going to have 46 pairs all on the same plate. Everything is on the same plate. But why am I showing it to you? And why do I show that we have 46 pairs on the same plate? Because once the microtubules on the both sides, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? 46 on this side, 46 microtubules, and the same for the other side. Imagine they are connected to the chromosomes in the middle. Why is it important? Because once we separate all the following structures, and once we cause the sister chromatids to dissociate from each other, and we are going to have double the amount of centrosomes, we are going to go from 46 chromosomes and 92 chromatids. We are going to go to 92 chromosomes and 92 chromatids. And 92 chromosomes are going to be divided between the two cells. Therefore, at the end, we are going to have 46 chromosomes again. So right now, how many DNA molecules we have in metaphase of somatic cell? We are going to have 92 DNA molecules. How many chromosomes are we going to have? Before anaphase, 46. While anaphase and after anaphase, 92. And when we do the cytokinesis and we do the cleavage forward and we have two new cells, 46 chromosomes in each cell. How many DNA molecules are we going to have in each cell after the cleavage forward? We are going to have 92 divided by 2. Therefore, we are going to have 46 DNA molecules in each new cell. Therefore, we are going to have 46 chromosomes in each new cell, because each DNA molecule, each chromatid is going to be separate. I hope this video makes sense. It's a bit stupid video, not gonna lie, but it's very, very, very important to understand this concept. Because when they give you only like the fly or something with 2 chromosomes or like 6 chromosomes, so we have, let's say we have like 3 paternal and 3 maternal. It's pretty hard to understand how they actually connect together and how they duplicate. It's more complicated to understand what is the process that happens in the fly cell comparing to the human cell. So in the human cell, it's way more complex, but now we actually understand how mitosis actually looks like when it comes to the human cell, that we're actually going to talk about when it comes to genetic diseases and many different things that are very important to understand when it comes to mitosis of the cell. So to sum everything up, mitosis in human cell. Before mitosis, before the S phase, we are going to have 46 separate DNA molecules. After S phase, we are going to have 92 DNA molecules because the paternal and the maternal got duplicated. Each DNA molecule got duplicated using our DNA polymerase and the process that we studied few lessons ago. Then we are going through prophase, through the M phase. We are going to see that the DNA material is going to be condensed into chromatids. The same chromatids, the DNA molecules of the paternal and the DNA molecules of the maternal, are going to come and join together through the centromere. Now we have one chromosome for paternal and one chromosome for the maternal, but each one will contain two DNA molecules, therefore two chromatids, therefore one chromosome because we have one centromere. But still, we have two DNA molecules instead of one that we had in the beginning. Then during the metaphase plate, all of the sister chromatids are going to align on the same plate. All 46 of them. Then we are going to take one of these, or all of them, and we are going to split them into two chromatids. The sister chromatids are going to get separated into both sides. Once we separate and we reach a point that we have 46 dissociated pairs that's starting to migrate to the opposite side of the pole, as soon as we break the bond between them, and we have double centromeres now because each chromosome contains one centromere, we are going to duplicate the amount of chromosomes we have. Because by definition, chromosomes equal the number of centromeres. So now, as soon as we break them, we have 96 chromosomes. And because we have 96 chromosomes, now the only thing that left is to create the cleavage furrow to break down the cell into two daughter cells and to split the 92 chromosomes equally between the two daughter cells and have 46 in each one, because 46 is the number of chromosomes we need to have in a somatic cell inside a human body. I hope this video made sense, and I will see you next time.